All right, now let us look at the structure, the infamous structure of DNA. Now, if you recall from the last spread, we clearly talked about DNA and told and confirmed that DNA is a double-stranded molecule and it is also a macromolecule and polymer. Macromolecule and polymer. So to form a polymer, we should actually have repeating units. So for example, to form a known polymer, we should have the same monomer repeatedly combining all over again. So what is that monomer for DNA? If you recall again, we clearly talked about how the monomer from DNA was the nucleotide. So DNA is a clear polymer of nucleotides. Okay, now we know the structure of a nucleotide. We know what a nucleotide looks like. So let us draw that out. 1, 2, 3, 4, 5. That's a pentagon or a pentose sugar, a 5-carbon molecule. This is the oxygen, and here 1, 2, 3, 4, and the 5th carbon lives somewhere here. 2, 3, 4, 5. Now I'm gonna get rid of those things to prevent any confusions and ISO. So boom, boom, boom. Right. Then I have a bond here going to the phosphate group. So let's draw the phosphate group as well. And then on this end of the spectrum, I have the nitrogenous base. Okay. That is the structure of a nucleotide, not DNA, okay? Just a single nucleotide. A single nucleotide. Okay, now we have to form a polymer of nucleotides. We have to form a polymer of nucleotides. So then how exactly does DNA form? How exactly do these monomers polymerize? Is it like this? Uh-huh. Does it polymerize like this, or maybe something like this? Does it polymerize like this? Or does it polymerize in a completely different manner? However, we need a polymer, no? We need a repeating thing. No, none of these diagrams show how DNA is formed. Or none of these diagrams show how the nucleotide is polymerized onto itself. There's a very clear, distinguishable method in which one nucleotide binds to the next nucleotide. So if I draw two nucleotides out like this, and I'm gonna form a bond right here. So that is how one nucleotide will bond to the next one. Like so. What happened to him? What? Okay. That is how the bonding occurs. Now let's duplicate that again. I'll get one more strand in right here. You see, a very important distinction to understand is this is not bonded to this carbon, okay? It's not bonded to this carbon, it's bonded to this specific carbon, like so. 1, 2, 3, 4. We'll just have 4 strands, like so. Now, if I zoom out, you can clearly see this is one singular DNA strand. This is one singular DNA strand. But we do recall that DNA is actually a double-stranded molecule. So where's the remaining strand? Now, I'm going to very roughly, this is a very rough structure, okay? This is a very rough case of what DNA looks like right here. I will be going into much more detail in a bit, but first of all, I have to give you the rough understanding. Now, this is what the second strand of DNA is going to look like. I'm going to have to take that, duplicate that like so, and we're going to have to do a little bit of rotation here. Now look closely. I'm going to rotate this strand like this. That strand is being rotated until it looks something like this. Hold up. Okay, something like this. And then I will be bonding that strand onto this strand just like this. This is a nitrogenous base, this is a nitrogenous base, and between those nitrogenous base, I'm going to have some form of bond. I won't tell you what exactly these bonds are yet. I will talk about them in a bit, and I won't also tell you why I have drawn 3 lines for certain parts and 2 lines for some other parts. All that information will be given in a moment. All you have to now understand is, now if I zoom out, this strand would keep elongating this way, this strand would keep elongating this way, and then still when it comes to all of this, what we're going to be doing is expanding in this exact manner, this direction and this direction, no difference. Okay, and here we have the intermediate bonds, thus forming the DNA ladder. I wanted to give you that outward appearance, like so. Nope, nope, don't get it. Now let's get rid of those guidelines. Right. This is the overall DNA structure. Now, this is a very vague structure. This is what's happening on the outside. Now, I want to dive into what's happening at the bonding level and give you much more detail. For that to be done, I have to get rid of these bonds like this, and then we'll draw in the carbon. So this is the 5th carbon. 1, 2, 3, 4, 5. 1, 2, 4, 5. There's an oxygen here. Alright, that's why the counting begins from this specific position. Even if you check glucose, if you remember from biological molecules, we started counting glucose from the exact same manner as well. There was an oxygen on the first point, I would say, of the hexagon. Right, this is the 5th carbon. I'm going to get rid of that 5 just because it's an iso and it might confuse you. Now, I'm going to draw the phosphate group in a little bit more detail. So thus far, we represented the phosphate group like this now, using a circle now. So I'm going to, let's reduce it a little bit like that. Draw what the phosphate group looks like molecularly. PO4 3 minus, no? So 1, 2, 3. And 4, and actually there are 3 minus charges on 3 of these oxygens. I'm just going to keep 1. You don't have to know about this in that much detail right now, right? It's not very important, but I want to show you how properly this bonds. That's going to give you a lot more context to this thing. Right, so I'm just going to get rid of the circle, right? And then I'm going to bring this in like this. And this is the first bond that's going to form between the sugar, between the carbon of the sugar and the phosphate group. And that is an ester bond. That is an ester bond. That's an ester bond. Okay, clear? Very simple. It's an ester bond. Then this phosphate group should also connect again to this above carbon. So this should again connect to this carbon. That is done in the exact same manner. We form simply another bond between the oxygen and the 3 carbon. So 1, 2, 3, no? The 3 carbon. That is also an ester bond. We don't have to go into too much detail why exactly it's an ester bond and not any other form of bond. Here, simply it's an ester bond. Okay. That is, now you see it's connected. Everything's connected. This is connected to this through the phosphate group, and this is connected to that. Here, you have noticed that we have 2 ester bonds. Usually, whenever we have 2, we can use the term di. If we have 3, we can use tri. If we have 1, we can use mono. Just like that. So this is actually, these bonds are together called a phospho. Because phosphate is involved, phosphodiester. Phospho 2 ester bond. Here we have a phosphodiester bond. Same deal here. This is also a phosphodiester bond. That is also a phosphodiester bond. Now, what exactly do these phosphodiester bonds do? phosphodiester bonds combine alternating deoxyribose sugars and phosphate groups. You can clearly see that is what is happening. Each and every sugar is linked to the phosphate group, then that phosphate group is linked back to the sugar. So that's how you form the singular strand of DNA going down. And that is exactly what's happening in each of these positions where I have indicated a blue bond. Now I can go ahead and draw all of that, but that's just going to confuse this entire structure and it's going to confuse the heck out of you as well. Just so you know, this is the exact same thing that's happening in every single phosphodiester bond between the sugar. So it's very important to understand. This is one sugar, this is one sugar. No? Between these guys, what we have is a phosphodiester bond. Not a glycosidic bond, nothing like that. These are in nature ester bonds, but we just don't call them just ester bonds. We call them phosphodiester bonds because of their nature. Two bonds, phosphodiester bonds. That's the first thing you have to understand, right? And then there's a second type of bonding in DNA, the green ones. The green ones. These are actually a type of bond known as, it's a very simple, very weak type of bond known as an H bond. H stands for hydrogen. This is actually a hydrogen bond, and you have hydrogen bonds between nitrogenous bases. Hydrogen bond between nitrogenous bases. The only thing hold, now you see, this phosphodiester bond is going to hold this molecule together. And then here we have the sugars. These molecules will be held together by a phosphodiester bond each as well. But the only thing that's forming this ladder, meaning this linkage, is the hydrogen. So they are equally as important, the hydrogen bonds. I told you they are a weak type of bonding, but just because they're weak doesn't mean they're not important. They are very important. And this bonding, this hydrogen bonding, happens in a complementary manner. We call this is the theory of complementary base pairing. Complementary base pairing. Remember how I told you that the four types of bases that DNA can have is adenine, guanine, cytosine, and thymine. These are the four types of bases. Now, there's a method to the madness. They can't. Now, if adenine wanted to befriend cytosine, unfortunately, that's not possible. Adenine doesn't want to bond with cytosine. So, adenine rejects cytosine. Now, cytosine's heartbroken, so he has no other option but to go and try for somebody else. He's going to try for guanine. So then adenine and thymine, they are suited for each other. So because of that, they will bond together. Guanine and cytosine, they also realize that they're perfect for each other. So they bond for life to each other. Something interesting you might notice. I have drawn two lines here but three lines here, which the same notion can be observed in the diagram itself as well. Some places have two bonds, some places have three bonds. Why have I done this and what's the theory behind it? It's very simple. Adenine to thymine will always bond using two hydrogen bonds. They are so friendly that just two bonds are enough to hold them together, while guanine and cytosine require three bonds, three hydrogen bonds, three hydrogen bonds to keep them together. So whenever you see a double bond, you should know it's possibly an adenine thymine bond. So we can fill that in right now. Adenine thymine This is a guanine cytosine thing. This is again an adenine thymine thing. But here's something people often overlook a lot. The thymine doesn't always have to be on that side. It can still be on this side. Still, that's an adenine thymine bond. Okay? And then I'll draw the same exact thing. Opposites. Okay? It's just, it has to be complementary. Adenine to thymine somehow. And there's no uracil here. Don't confuse anything yet. There's no uracil here. Uracil only comes when it comes to what? RNA. This is DNA structure. Deoxyribonucleic acid structure. Right. Those things. And you see, once the bonding is complete in all sorts of manners, we get the DNA double-stranded helix. Double-stranded helix. First of all, one strand, second strand, and then the hydrogen bonds form the ladder. It's not just a double-stranded molecule, it's a double-stranded helix. What does that mean? That means it's actually a twisted helix. And that is how this structure is born, and this is our modern DNA. This is actually known as the Watson and Crick model of DNA. Your syllabus does not require that you know that much information. If you are interested, do give a small Google search. They're good people, I think. I did not know them personally, but that's what history is saying. The Watson and Crick model of DNA explains complementary base pairing and how phosphodiester bond formation produces this ultimate structure. One last small tiny detail I should add to this is the ends of DNA, the ending. It's not a hard thing, it's a very simple thing. I'm just trying to find a color that was not used before. Orange, yeah, that's good. Okay. Focus on this. Remember how we termed the carbons that was 1, 2, 3, 4, and the fifth one lived somewhere here, right? So if I consider this end of DNA, this is actually the 3 end, right? We usually call this the 3' end, meaning 3' end. We call this the 3'. Now, if you know from in some form of perspective, you know how to solve a Rubik's Cube. I think you know what I'm talking about, a Rubik's Cube. You know, we have the term prime, and we usually denote it like this. That means to go in the opposite direction. So this is just 3'. This is the 3' end. The dash means prime. Okay, now I'm going to go all the way to this. Now here, let's number the carbons 1, 2, 3, 4. And the fifth one lives somewhere else, somewhere here. And from this end, you can see the exposed end is the 5' end. The exposed end is the 5' end. So clearly here, we're going from the 3' end all the way to the 5' end. We're going from the 3' end all the way to the 5' end. If you look at the other strand, if you look at the complementary strand, you can see it's the exact same thing, but now 1, 2, the third carbon is here, so that is the 3' end. The fifth carbon here is open, and that is the 5' end. And we're going from this to this direction. Do you see? We're going from this to this direction. What can you get from this? One strand is running this way, the other strand is running this way. The noteworthy mention you gotta get out of this is DNA is referred to as an anti-parallel molecule. We have 2 anti-parallel strands running. We have 2 anti-parallel strands running. And thus we have the overall structure of DNA with the utmost amount of details that you are going to need for this level. Just a small recap until we hit the 20-minute mark. DNA is a polymorph nucleotides. We have two antiparallel strands running from 3' to 5'. On either side, we have hydrogen bonds in between nitrogenous bases, and each sugar is linked to the other via a phosphodiester bond. If you understand these things, you can build your own DNA. You can write down yourself how each and every point is linked to the other to bring us this infamous molecule that we all have very tightly packed inside our cell nuclei. The end.